Hello, everyone. Thank you for joining Cota Access today. I'm very pleased to have a chance to speak with Dr. Chun-Hao Huang, who is the CEO of Algen Biotechnologies, one of Cota Capital's portfolio companies. First off, Chun-Hao, congratulations on being one of the recognized companies at the JP Morgan Innovation Summit in Dubai just last month. So good for you. So why don't you start off by telling the listeners about Algen Biotechnologies?
Sure. First, thank you, Janis, as well as the Cota Capital team for this great opportunity to speak here. So also, I would like to say hello to all the attendees online. Thank you for taking the time to learn more about Algen. So Algen is a company that originated from Nobel Laureate Professor Janis lab at UC Berkeley. And our mission is to revolutionize therapeutic drug discovery through our advanced CRISPR gene modulation and AI. And our long-term vision is to become the central drug discovery and development engine to enable the next generation data-driven pharmaceutical industry. Yeah, so that's why in the past few years, actually our team, with the support from our investor, including Viking Global, and of course, Cota Capital, Team Jeffers, Associate, Illumina, and Y Combinator, we are able to use our strong team to really focus on our proprietary CRISPR-driven data creation and learning at scale to find new treatment for important diseases such as cancer, inflammation, and metabolic diseases. And also, not only we have a very strong foundational platform, we are actually very quickly enable discovery of new biology based in class molecule, allow us to quickly get to small molecule candidates to enable the potential new therapies for oncologies, as well as we work with large pharma companies such as Pfizer. We are tackling the most important inflammatory disease indications, as well as with Novo Nordisk for metabolic diseases. And so I would say definitely it's a great honor, as you mentioned. We are recognized by the JP Morgan Life Science team, Blue Horizon Advisors, as well as the really international world-class judge of panel led by Dr. Glimcher, who is the Dana-Farber CEO. So there definitely is a great proof of our Algen's achievement, progress, as well as really good demonstration of the company trajectory on the right direction. Yeah.
Yeah, no, that was a very notable meeting. First of all, it was the annual. It was the kind of the inaugural summit for the JP Morgan event there in Dubai, but also the two winners. Algen got an honorable mention, and the winner was another company. Both of them were really AI based, and I thought that was pretty notable. But congratulations again on that. Can you tell us a little bit about yourself, Chun-Hao, kind of your background and kind of your journey in terms of founding Algen?
Sure. It's a long story, but I will talk in a very concise way.
We got time.
Yeah. So I would say I'm a scientist by training, and I think everything's actually happened when I was in college. So at that time, it's actually the time that Human Genome Project is completed. So around 92% of the human genome actually gets sequenced. And so back then, I read a really good book that impacted me a lot. That's written by Kevin Davis. It's called Cracking the Genome and Unlock the Human DNA. And so through that book, I got 3 take-home messages that still become the root of what I'm building up, right? So the first one is actually talk about innovative technology. To be able to achieve such a huge, impactful project, right, for human beings, we need to develop really innovative technology. And one part is on the wet lab, right? Mm-hmm. Human genome is so huge. How can we actually sequence it, right? So we need to develop technology like shotgun sequencing. We chop the human DNA to different fragment, sequencing it all. On the dry lab side, then we also need a really sophisticated algorithm, right, to be able to map out all these individual fragment DNA sequence back to the human genome. So that's
I'm going to make you stop for a moment because we have a great number of listeners. You and I use the jargon of wet lab, dry lab. Would you mind just kind of explaining what that means a little bit?
Yeah, So Wet Lab is our bench scientists, right? They do day-to-day great work on the bench to actually do cellular experiments, right? Molecular biologies. So it's actually generating a lot of data to understand the biology of the cells. And the dry lab is the data team, right? So basically, data scientists curated the data, running the data through pipeline, and actually decode the meanings, right, of what generating from the wet lab. Yeah.
Good, good. Okay, great. So you can go
Yeah.
back to your combining of the both and the dry lab.
Sure. And then that's the first part about we need an innovative technology. And second part is the data, right? From this technology, you're able to create a really meaningful data, such as decoding the human genome sequence, right? And that enable a lot of new discoveries through that. And the third piece of about that book I learned is we can actually create a business or we use business venture to accelerate this process, right? So we can actually leverage the sequence to be able to understand human genome as well as develop new therapeutics. Yeah, so that's, I would say, the origin of my career journey. And then the second point is when almost a decade ago, I was able to join a conference called Lindau Nobel Laureate meeting. So there I met Dr. Michael Bishop, who actually discovered the first ever oncogene mechanism.
Yeah.
So there I was discussed with Dr. Bishop about how we can actually enable, right, future medicine as well as discover new therapeutics. And then we nailed down to two main topics. I think the first one is about the big data, but we need to really generate meaningful big data to be able to recapitulate the human disease progression. It's not just a data with low quality or no meaning, right? It's actually need to be human-centric, patient relevant. Second is we need to have a really best technology, functional genomics technology, allow us to decode the causal pathway, because understand progression is the first thing, right? Be able to understand the causal pathway that driving this progression, then that become the central thing allows to enable the new discovery of therapeutics. So those two become, my route together, and through the journey, I think one thing very important is to have great mentors. So I was very grateful through my PhD with Howard Hughes investigator, Dr. Scalo, Dr. Chapshers. They are the leaders in functional genomics area. So there even before CRISPR, I already built on RNA technology to do the drug screening. And then, of course, most importantly, to be able to work with my postdoc mentor, Professor Doudna. So there, we really start to put in CRISPR and AI together to formalize and make the Algen vision to become a reality. And I would say, definitely our goal there is to use CRISPR in a different way, right? Other than other companies, to be able to generate a really meaningful, massive data, allow us to keep learning the biology, and that enable the new discovery and create a medicine for patients.
So if I can kind of paraphrase a bit, because you gave a lot of information there, and then also kind of add my own experience, having kind of been in, gosh, let's see, I finished my PhD in 1991, so it was a while ago. So certainly in the period of sequencing the genome, collecting all that data, the idea was that we'd be finding insights into diseases by having all that data, and certainly. And at the time, it was very expensive to create that data. So as time progressed, the cost of sequencing came down. The human genome and others got sequenced. We all learned that didn't provide all the insights we needed. So you needed something more applied. And so ways to intervene or annotate all that data in the human genome. So as you were progressing through your PhD and then also with your postdoc, it sounds like you were able to bring that, the whole point is bringing together information that informs each other's steps. And so you've got ways to look at really focusing on human data, because that gets you closer to human disease than the traditional way of looking at mice and rats and non-human primates. And then the additional application is, as you look at new targets in these pathways, you're really able to test it quickly early on using the CRISPR-based technology. So maybe you can go into a little bit more detail around those two aspects of bringing it together, as you kind of, joined, Jennifer Doudna's lab with the CRISPR piece. How did you kind of think about putting those two things together? And if you want to talk a little bit about your two platforms, with Algen Brain and Algen Discovery. So maybe talk a little bit about that.
Sure. And first, I want to echo you on the completion of the Human Genome Project, right? Definitely, there's a lot of hope about we can probably solve a lot of disease, right? But at the end of the day, there's still a lot of unmet need because we have the DNA sequence, but we don't actually understand the gene functions, right? So that's why a functional genomics way to actually identify and understand the gene functions, right? That's more like the current frontiers, and that's what we build on. And yeah, quick overview about our Algen's technology. So I would say, as I mentioned, right, understand disease progressions and be able to further interrogate, right, the candidate genes. That's the key central things of what we are building at Algen. And we have a foundational CRISPR platform allow us to achieve the best in-class gene modulation. And so to do that, we actually make several innovations. So we re-engineer Cas proteins to achieve those, as well as we modify the second important element, the guide RNA scaffold. And third is we even generate a proprietary CRISPR gene modulation data, allow us to further inform how we can design the best guide to modulate the genes. And so far, I'm very proud of our team that we are able to do head-to-head comparison with existing gene modulation type of technology. So we are able to outperform the current standard of methodology. And for most of the genes, we are able to achieve 99% gene inhibitions, 20,000 folds gene activation without cutting DNA. So one part we are also separate from the traditional Cas9 system is we don't cut DNA, we don't induce double-strand DNA break. So that's why we are able to have a very reproducible result without any bias, right? Because we all know cutting DNA will induce DNA damage response, and also there's a bias about the chromosome rearrangement and so on. So that's on the Algen CRISPR part. So that allows us to achieve the best gene modulation for drug discovery. On the AI side, we have our foundational data universe that cover both oncology, inflammation, metabolic diseases. So allows us to build a human cell type landscape, right? And through that, our proprietary model can identify the disease trajectory all the way from healthy to intermediate stage and to later stage. And so one part, the reason these particular angles can solve the unmet need is the current way that pharma or some like public or later stage company, when they look at disease, it's very binary, right? Always talk about healthy disease, but regardless how many samples you're actually collecting, right? But we all know disease progression takes decades, right? All the way from healthy cells, intermediate stage to the late disease. So that's why we build these trajectory models, allow us to map out that. And on top of that, we identify the causal candidate genes following this kinetic, and that become our CRISPR functional validation candidates to de-risk the drug discovery.
Yeah, I think from a Cota capital perspective, when Cota first invested in the company, I think what was so interesting to us is this really combined approach of the CRISPR intervention, right, and the optimization that goes around the succeeding generations of the enzyme, the technologies, but also bringing in the analytics piece, right, and combining the two, because you're able to, as you set out and described, you're able to first of all start with human data, human cells, and then look at it in a non-binary fashion, but in kind of a multifactorial, multi-genetic expression pathway of progression from healthy to diseased. And maybe you can talk about it in very soon, is it even in the area of oncology, right? a normal cell doesn't immediately become a cancer cell. Lots of things go on within that cell to turn it into a cancer cell. And I think that's another really interesting thing about your technology, and then also the capabilities that you've developed, is that you're looking at this not only as a progression as cells change, but you're looking at the population of cells in total, but also at the single cell level, these changes. And through your analytics, you're identifying really new and novel targets in a pathway of disease. Can you maybe, talk about that a little bit more as to kind of, how you came about to say, hey, look, this is the better way to intervene to look for new targets and new discovery?
Mm-hmm. Yeah, so definitely there's multiple layer of how we actually discover, right? So first part is on new target discovery. And so as I mentioned, our algorithm is actually based on human-centric data, allow us to map out the disease trajectory. And it's actually learning really unbiased based on the single cell level RNA, non-coding RNA informations. And then we can first identify all the possible paths that toward the disease, right? And in some cases, not only all the way from healthy to disease, we also model the treatment sensitive all the way to treatment resistant. Because there is a huge market, also a many for patients. It's like the current marketed drug, right? It's have good effect, but again, right, the cancer resistance or some effect really evolve, right? Relapse comes in. So can we actually have a technology to identify a potential combinatorial therapy, right? To enable and even strengthen the current therapy. So there's a huge market surrounding that. And then by identifying these candidate genes, our CRISPR way, we actually implement CRISPR into individual single cells. And we can modify and track all these CRISPR modulations, how that changes based on billions of RNA data points. You change one genes, you change all the other genes, RNA expressions, as well as our technology is really capable of doing combinatorial gene modulation. So for example, gene A, 90% separations, and then gene B, 24 activation, right? So all that allows to actually decode a pathway. And one thing I want to mention, so that's on the new target discovery, we actually have a follow-on technology can allow us to quickly get to a drug. The magic there is we actually use CRISPR target fingerprint as a standard. And then by doing that, we all know that every drug have their off target, right? So we use CRISPR to create how a good drug should look like on target, less off target. And then we use that to quickly screen small molecules, some mature modality, and that allows to get to drug faster and for development.
Yeah, I think certainly the faster component is very compelling in terms of transforming drug discovery. Having spent a number of my years in biotech and pharma, time is money, so fast is good, IP life and such. But I think also what is so compelling now with the combined technologies is not only can you maybe reduce the time it takes to get to the drug that you want to put into the clinic, but also hopefully we want to increase the probability of success of getting through the clinic. And you bring up some very good points. And, for those on the call that are interested, but maybe not, true aficionados in this space, is you mentioned off-target effects. So when you say on target, it's like you develop a drug that hits a specific target that gives you a specific response, and that's efficacy, and you want to get that. If it's off target, it could lend itself to some sort of adverse event or some sort of side effect that you don't want, and that can kill a drug. So if you're able to do a lot of the kind of in parallel answering of questions within, it's a human pathway, it's human cells, human pathway, and that through CRISPR, if you intervene, you can make some changes. And then also you could predict potential off-target effects and avoid those and choose your molecules accordingly. Not only are you increasing speed, but you're also hopefully coming up with a higher probability of success. And that definitely is hugely meaningful.
That's right. And one point on that is we actually build in a toxicity phenotype on both target screening and drug screening. So we use CRISPR to suppress, let's say, essential genes, right, for replications. and that literally become very valuable to de-risk all the downstream drug discovery at the earlier time.
Definitely. And I guess you can, we can kind of sense our excitement about this space. I think as an investor, what was really exciting and compelling to me is that you guys have actually reduced your platform to practice. And how I kind of see it is we see a lot of fabulous technologies generated by super smart people coming at us saying, hey, look, we've developed this and we think it can do X, Y, and Z, right? And usually initial funding, seed funding goes to kind of establish that. What's fantastic about Algen is that you guys have reduced this to practice, right? You've done a nice job of explaining a very complex platform and system. But, as they say, the proof is in the pudding and the eating, is that you actually have, I would say, kind of, preclinical molecules for specific applications that came out of your platform in a couple different ways, but for a couple different purposes. But can you just speak to that so people can kind of bring this back down to, what does this mean, in terms of ultimately what a patient would get as a therapeutic?
Mm-hmm. Yeah. So I would say when we start Algen, right, our goal is definitely get the good medicine to patient as quickly as we can, right? So that's why not only we have the platform built in an advanced way, we also have a really strong teams on oncology, especially our CSO, Dr. Jeff Wheaton, who is a really senior drug developer, has been developed FDA-approved drug, which approved by FDA last year. And so Jeff bringing a strong drug discovery as well as oncology expertise, as well as our advisor, Dr. Frank McCormick, really pioneers on the RAS pathway oncology and drug development. So with their oncology expertise as well as my functional genomics insights, so we are able to really using our platform in two ways, right? First is, can we identify novel targets that allow us to conquer, let's say, all the RAS mutated type of cancer. So we all know RAS is very important, and now there's a drug can directly target it, right? In particular specific mutation, and a great drug from Amgen Marathi. But again, cancer cells are smart. They come back with different mutations or resistance very quickly, right? So what Algen, our approach is, we actually look at all the pathways that are driven by different RAS mutations, but even different RAS, right? So can we find a weakness, common weakness around this pathway, the hidden link between RAS and the pathway, right? So that's the new biology and targets we are focusing on develop our new molecule. And the second part is regarding if a drug, right, a targets that in the clinic that it's very hard to have a selective inhibitor, right? Using our Algen's drug screening approach to have on target and less of target fingerprint, right? That also allows to can get to a development candidate very quickly for one of our program for MYC-driven type of cancer. Yeah, so these are the two ways we really reduce to practice, right? From the platform all the way to the drug candidate with our strong team and platform.
Yeah, you bring up a good point when you mentioned about building out the team and the experienced discoverers. I mean, that's I think where we really see the most advancement or the opportunity for advancement is taking the young innovators like yourself who are really experts in developing new technologies and platforms and applying them, and then really marrying it up with very experienced people in specific domains, such as Dr. Frank McCormick and the RAS pathway. There's probably no better expert in that space. But then also Jeff Witten, who has spent years, various pharma companies and biotech companies that know how to, because this is just one part of it, to bring these molecules forward and actually get them, through development and eventually to the market. So I think that is a key strength that Algen did early on, right? I mean, you guys are, just finishing off your seed financing and looking to raise some more funds. So I think it's that really bringing those pieces together and recognizing that there's a need that goes along with moving from high tech innovation to actually, getting therapeutics through development. So that brings us to kind of like, what's the next step for Algen?
Yeah, so you mentioned we actually have a very capital efficient way to use our seed funding, right? So for the audience, we actually achieved, let's say, platform innovations on proprietary CRISPR system as well as our AI models. And also in the meantime, we are actually to develop our internal oncology pipeline and get to a development candidate as well as a lead molecule for MYC and RAS-driven type of cancer that account for like 30–50% of all human cancer populations. Yeah, and so for Series A, we are looking forward to advance our pipeline as well as our platform, and also we have ongoing partnership with large global companies, and definitely we are expect to make extension of those to a larger deal, multi-years deal with our CBO, Dr. Kristen too. And then I think through that Series A raise will allow Algen to get to the next phase that toward the transition to become the clinical stage companies. And I think one thing I want to mention is at the early days, right, when I sit down with our investor, Viking Global and Illumina, so we definitely understand the value of the drug, right? So that's why when we initially started Algen, we know we are going to push the drug forward to certain inflection points. And then later on, we can decide to co-work with the pharma companies, right? Yeah, so that's on oncology side. And beyond oncology, then we definitely welcome the pharma companies like right now to work with us for drug discovery.
Yeah, and I'm going to throw you a little curveball on this one. So you've applied it in the area of oncology right now with your molecules. You are exploring applying it in inflammation and immunology. And I think really, do you think there's an even much broader trajectory in terms of applying this sort of platform, especially around, diseases that are multifactorial, right? Not monogenic, but, lots of different pathways. And one that comes to mind is in the area of neuro or like schizophrenia. So if you can get, do you foresee this, that you can explore this space either as Algen or others in terms of really going after some of these intractable, really complex types of diseases?
Yeah, totally. I think for the disease, if there's no known drivers, right?
Yeah.
That's actually that's actually a good indication to demonstrate our strong capability. So on oncology, one part is there's most of the time there's a defined drivers, and the reason I think Pfizer or Novo Nordisk, right, they love our platform, fall in love with our platform is because disease like inflammations,
Yeah,
metabolic diseases, or neuroscience, right, neurodegenerative, right, there's just no clear drivers, and we really need a way to decode, right, these causal pathways. So definitely, I think there's a great potential also on the neurodegeneration side, and we will do that step by step, right, to actually conquer the disease.
So Chun-Hao, I think, hopefully there's some more entrepreneurs, listening to this webinar. So you've learned a lot in a very short period of time, being a founding CEO, coming out of some very notable labs, excuse me. But, as you've had this accelerated course, can you just, touch on a couple things where you're like, look, this was very transformational learning for me as I, expanded my career this way.
Yeah, sure. I would say transition from a scientist to an entrepreneur, there's definitely a lot of learning, right? But also there's some common things between the scientist and the entrepreneur, right? It's like we are definitely goal-oriented and we work so hard, on different domains, right? And also we are solving problems for people. So there's some common share things. And however, I would say not only myself, but also my teams, right, when they join Algen, one thing we really want to do is we want to make impact, right, to the patients, right? At academia, we do a research, and then we really want to see how we can actually evolve from that research to actually make a better drug for patients. So every time when I interview our team members, definitely I think that's one part. The outstanding scientists, when they join us, right, they bring up about can we actually make impact, right? So I think that's one thing. And then the other thing I really appreciate Cota is I think under Bobby and your guidance, right? Because Cota is a multi-stage investment firm, right? So all the way from earlier stage to public company. So really, I think through the interaction with Cota, I was able to learn a lot of like a big corp type of the way how to run a company, right? And also we are facing enterprise type of the collaborations also with government as well. So I think great learnings and also great guidance.
It definitely, it takes a village, as they say. So this is bringing us to a close. And so I do need to show a disclaimer slide, which I'm going to try and pull up in a moment. But as, you know, perhaps you can do a little voiceover as I'm doing that. And why don't you tell us kind of, you know, what you do for, what do you do for fun and relaxation?
Yeah, I would say major, I think two things. One is, since my childhood, I really enjoy reading detective novels, and I'm a big fan of Sherlock. Yeah, so that actually is a good, you know, insight when now I'm finding a new drug, right? Because you need to observe a lot of informations and digest those information to be able to find a subtle, important things, right? To make some important decision. So I think that's one part really enable from my childhood habit now to drug discovery. And second, yeah, I love watching sports, all kinds of sports like basketball, soccer, baseball, and then I think one of my favorite is Formula One. So I really enjoy, you know, a team, right? Work together. Let's say in a pit crew, right? You only have a couple milliseconds. All your team members from all different expertise, right? Engineer, software, you need to come together to make the decision, right? And everyone doing the things toward the same goal, right? To winning the race, right? The champion. So definitely I can see there's a great spirit there, can be leveraged for biotech industry as well. And hopefully, yeah, with Algen, we can definitely be able to leverage that and then to create like most impactful medicine for patients. That's the every day we wake up is trying to do that. Yeah.
That is definitely a commonality within this business. And it's quite interesting, your you know, interest in detective stories, as I love those too. And I always did love Sherlock as well. But it does seem like even your rest and relaxation does seem to, you know, also, you know, be in concert with what you do as your vocation. So anyway, I do want to thank you, Chun-Hao. It's been a delight. I know I've been working with you on your board for the last couple of years, but certainly I'm continue to look forward to the progress that you and the team at Algen is making. But thank you very much.
Thank you, Janis, and thank you, the Cota Capital team. Thank you.
And
Cheers.
thanks everyone for joining.
Thanks, everyone.